Hi, I'm Oscar Istis, a student at the University of Kentucky. I'm helping to make the text for this project and compile it all into a movie.
I'm Abigail Greenhalgh, an undergraduate at the University of Kentucky, and I'm working with Oscar to draft text for the project and help compile a movie.
Hi, my name is Becky Kroll, and I am a faculty member in the STEM Education Department at the University of Kentucky. I am working with a team on developing the overall modules, providing scaffolding for educational support and student learning, and developing the story theme that goes along with our module.
Hi, my name is Jay Bernard, and I'm working on the field simulations of this module.
Hi, I'm Erin Richard, and I am helping to make laboratory simulations with the bacteria cultures and also test preferences of the insects to egg laying and diet choices in this project.
Hi, I'm Robin Cooper at the University of Kentucky. We are taking parts of this workshop and developing it for undergraduate research projects.
Hi, my name is Tawny Aguayo Williams, and I'm a high school biology teacher. I'm beta testing this module for a class project, and will provide feedback on how that goes.
Now that you've had an introduction of all the people that are participating in this module, so the idea is this forensic case. Person's on the grass, apple's been bitten into, and the kind of information that could be collected for the students or given to the students to then work out the case. So we have this very flexible, as I mentioned, but to actually have some outline to the project so the students don't just go all over the place. There are different tasks, and some of the tasks listed down here, like 4 and 5, are more detailed that might be for a classroom that has situations such as you can monitor developmental stages, or you have a fume hood where you might want to grow larvae in, or even culture dishes. Or if you have a field that you can work in which won't be disturbed by animals, then you could use maybe optional task 4s and 5s. So we'll go over these different tasks here, and I think this is laid out nice that can be worked on. Task 1 is that the students could then collect information such as body temperature, condition of the corpse, is the skin broken or intact, what type of insect larvae are around, and then think about the environment, the plants, the grass, and if insects were present at all the different stages, for example, all the way to a pupa, and do we have pupa cases? Then also look at the apple. Is it dried out or moist, bacteria, fungi growing on it? What type of insects are here? Do we see blowflies also on the apple, or only fruit flies on the apple, and vice versa for the body? Are fruit flies seen on the body, for example? And then the types of environmental conditions can be varied under the whatever the teacher wishes to maybe pose for the students, because that can change the developmental time of the insect larvae. Task 2 is to do Google searches on these two insect species, one, the Drosophila and the blowflies, and they can actually find out, oh, how long does it take to develop from an egg to a pupa to an adult, for example, in Drosophila, and is it the same for a blowfly, and under what temperature conditions? And then thinking about maybe other insects as well. But then you can back calculate to determine how long potentially the body and the fruit was there on the grass when it was found. Task 3 can be to actually take that information that was gathered and looked up online and then list that all out, like list out the different types of larvae in the eggs and the pupa cases, and then try to come up with a developmental curve based on the temperatures that the teacher so wished to give to the students, for example. And one then can calculate the time potentially for the person that have died or the apple that's there. We can use some NetLogo simulations. This is freeware for the students. We found in the past the students really liked playing with NetLogo software. We have a website here, and that you can download the NetLogo software and some different modules. you can then click on Module 1 or Module 2, Module 3, and you can download the files that will then run on the software. Task 4 is to go over how to set up and simulate the scene with mixed fly species enabled to monitor the food sources in insect development. Task 5 could be something of going over to set up the individual fly species and say effects on development. So one could take Drosophila, for example, put that on an apple, and then inside of a fume hood or closed, semi-closed conditions with like screen, as I'll show you, then look at how long does it take the Drosophila to go from egg to pupa, and then eclose as an adult under those defined temperature conditions, for example. One can even mimic this as it goes hot in the day and colder at night if you have an incubator, for example. So there's a lot of different variabilities that one can mimic. With this task 4, you can actually have a cage. If you're going to simulate this, if you're going to put it outside, you might want to have a cage that you can put the meat or the apple into, for example, so you can bring it indoors at night. Or if you're going to put it outside, be leery of wild animals that can come in and take away your project. And so you might want to use wire around it, or a cage, for example, that will not be taken away by a wild animal. But it might be a more natural condition if you want to simulate it out in, say, the environment around that school, for example. So for simulating, what we've done in the lab, which worked out really nice, is to choose beef liver for the blowflies, because they do not tend to do well on the apples by themselves. And then for the fruit flies, we took pieces of apple. And then you can look at the egg laying, how long does it take for the eggs to start to be laid in the conditions, and how many over the certain days. If one wants to, you can be very defined on these conditions if you want to simulate it. What we did here is just took some plexiglass, basically some glue with a hot glue gun, and then tape to help hold it together. Tape to help hold it together. And then we have a hole here that we put a screen, a very fine screen on, and just taped it onto the side. And you can see that the other sides are closed. What we did here was actually just take another hole and glue a wooden ring around this, and then take an old pillow cloth and cut out one side that would fit over the ring. So this is a wooden object that you can get at a craft store for knitting or crocheting, and then glue that to the wood, and then your pillowcase can fit on top of that with a clamp. And then on the inside, we have a number of flies that we introduced to the inside. We have beef liver here and an apple, and then some extra water to keep them hydrated. So here's the fresh beef liver, and here's the apple, and a close-up of it. For example, what's nice about this kind of cage condition is you can reach in through the pillowcase, and the flies won't all come out, and then take this container out and then look at it closer with a magnifying glass, for example. A hand magnifying glass works very well. But now we can see the flies. They're laying eggs on the food, on the apple and the liver. And then they tend to get water from the hydration station here we have. You have to keep the flies hydrated. If the apple and the liver dry up, then the flies will die up relatively quickly as well. But that could be part of your simulation. Okay, so here's you see the number of flies, and we have some dates up here when we actually collected these pictures, and we can see how it's changing over the time period. And now we see eggs over here on this side. These are Drosophila eggs with these two little spiracles that come out. These are blowfly eggs, and you can see they're quite different in their shape, these long structures without those two little spiracles. And now we see that they're laying lots of eggs. So we see eggs all throughout on the liver as well as on the apple. And if we look on the apple, the fruit flies have laid a tremendous number of eggs on the apple. Very few blowfly larvae on the apple. More of the blowfly eggs are on the liver. And now we can see these eggs next to each other. Here is a Drosophila egg with those two little horns, and here is a blowfly egg. So the size difference is very easy to compare. Then we can start to look at when they close and become first instars and start crawling around. What we see here is we see some instars crawling. We see some eggs still. And now we can actually see quite a mix of information here, where we're still getting eggs, we're getting first instars. The eggs of both the blowflies and the Drosophila are hatching. And then continue to look at it over time, take more pictures, document it. Now we see this is 410. We're having quite a number of larvae crawling around. And here's a little movie. You see the different stages now. This is a smaller one, this is a larger one. These are both Drosophila. In that case, they actually crawled over to the beef liver, which was interesting. Okay, and to simulate this, this was actually in my back porch on a table here, and you can just tie off that pillowcase and other flies don't get in, but you don't want to have it. Maybe you do, you know, depending on the environment, but it can get quite hot inside this cage. So we had it under a semi shelter, but also to protect it all from the rain. Okay, and now we can see at a later time, look at the number of eggs and larvae that are all over this apple, quite a bit. Okay, and now we have at, say, the time that you want to take a snapshot. Maybe this is the time you find the body and you gather up all your information. So you'll find eggs of the different species, you'll find the larval stages, and now you have to find out which one's Drosophila and which one's blowflies. But you might also find pupa cases. So here are the two different types of pupa case. Here's the blowfly pupa case, here's the Drosophila pupa case. Quite different. But also what to look for very carefully are the empty egg cases. That means, you know, they've hatched out, but also the empty pupa cases. So here's a pupa case with a Drosophila still inside. These are the eyes developing of the adult. This pupa case, the adult has already come out. There'll be a crack on the edge of the pupa case and the adult has emerged. So now we know that this must be a longer period of time, because how long did it take for that adult to develop in the pupa, right, under different temperatures. And then some extra photos that we've taken over the time period. One could simulate, say, a rain that came by and washed away a lot of your larvae, but you had empty pupa cases and maybe empty egg cases around. In this case, we actually see mold growing on some of the pupa cases, and so that would take even a longer period of time. And these are empty pupa cases, so one can actually look at more information. Online, one will find a number of different websites with cycles of, say, blowflies or different types of insects. And note that it depends on the developmental temperature, or the temperature for the developmental curve. Higher temperatures usually develop faster, lower temperatures slower. But if you have day and night cycles, that could impact this as well. So there's a lot to consider in this life cycle of the different insects. And for the blowflies, we can see, you know, from the eggs to first, second, third instar pupa into the adult, same like with Drosophila. One might find different pictures online. These are two different ones I just found on the website. This one doesn't give you the time cycle so much. This one does give you the time cycle for room temperature, 21°C, for example. And so one will find different types of information on the website. Now, to really stage your larvae, one can look at the length of the larvae and put them on a micrometer microscope slide and actually take pictures with your iPhone through the eyepiece, and you can see how large your larvae is. But depending on the food, Some larvae will be actually smaller if they're not getting good food, like in the apples all dried up, for example. You might still have a third instar larvae, but it would be as small as a second instar if it was on the fresh apple. So length sometimes is not the best indication of the developmental stage. What people use to be more careful with this is actually the mouth hooks. So the mouth hooks in these different stages right here, you can pull the larvae apart, and you can actually take the mouth hook and put it on a slide and then a cover slide, and then glue that cover slide down, and then look under a high-powered magnifying lens, say a compound microscope really, and maybe a 20 to 40x objective lens and a 10x eyepiece, and you can see the teeth that are on the mouth hooks. The number of teeth or the structure of the mouth hook tells you if the animal has molted in between. And so first instar only has like one hook, second instar has like two, but a very sharp little bump there, and then the third has multiple mouth hooks on there, bumps on the mouth hook, and then you can tell that that was truly say a third instar. Task 5 can be to simulate this in a laboratory environment under given conditions, such as putting them in a dish, a little petri dish. The problem is if you put them in a petri dish, like fruit flies and some food, and you close this down, the CO2 can build up in here and kill your flies out. So what we've done is to make a hole in here with a soldering iron, and then glue nylon mesh on top of it so it lets the air in and out. But also you can add water through this mesh and keep the food moist, for example, and you don't have to open this up. You can also put plastic around the edge to keep them from crawling out. If you're looking at the larvae, sometimes they can go up and out. One can actually use an old aquarium as well and put different food in there. You can time it to see, different times, where do the adults go to, where do they lay the eggs, for example, on different types of fruit, some different conditions if somebody wished to test that out. And then if you had an incubator, you can vary the temperature day and night cycle to simulate, say, the conditions that one might find out in the environment. With airing, we're actually working on some other aspects in looking at bacterial endotoxins, particularly gram-negative bacteria called lipopolysaccharides, LPS. This is more an advanced type of an experiment. I'm not sure that this is available for high school labs, but we're using this in the college lab, and we're actually seeing if larvae can tell the difference with food sources without gram-negative bacteria and with gram-negative bacteria, and seeing if they'll prefer one side over the other, but then looking at active or particular ingredients that they might be sensing. And it's thought that insects can actually sense the lipopolysaccharides in gram-negative bacteria, and this is how they choose the different environments to lay their eggs. One can choose without and with, or different concentrations. One can also look at the adults and see if different levels of food, or if the larvae will choose, if they were in the food, would they choose one side over the other? So do adults and larvae both have preferences? So this is where we're going at, say, a more advanced level of investigation. There's also, all these projects are on the website. We also have a project where the students, you could give them a project at home with a paper and say a web link for an engaging activity. If they can't come to the class, you could give them a homework assignment or maybe an extra assignment. And this is kind of fun. We've actually have some middle school kids participating on this now, where we give them the scene, the information such as this, and tell them that this is an information that has been gathered. We give them pictures of the larval Drosophila at different stages, as well as the blowflies, with or without pupa, for example, with or without pupa that have eclosed, that have come out, and then ask them if they can figure out what time of conditions when this had occurred, and the apple is on the grass and the person's on the ground as well. And then they've looked up this information. We can actually give them this information, or you can let them look it up. Depends on the level. So what we had here is we actually made a key with all the different items that were collected by these two different types of insects, the blowflies as well as the Drosophila, and maybe some extra information like empty egg cases. Did it rain? Did it not rain? And here are the closed case here and a closed case here in the pupa of blowflies or Drosophila. And then that the teachers can mix up these content for the students as they wish and give them different information. So here's an example that we gave. We have, all these different stages, and then the students can work that out on the bag of information that they get. They just put everything out on the table and then have to back calculate to the time periods. So for the teachers, we have on the website a PowerPoint that they can download with all these items, and they can cut it out and put as many on the bag that they want and simulate this. And so here's different flies, maybe the Drosophila versus the blowflies. We have also on the website all the pictures of the eggs, so you can make multiple copies of this, of the blowflies, for example, and different stages, like the first instar of the blowflies, or the second instar of the blowflies, or the third instar of the blowflies, or the later third instar of the blowflies, all the way to the pupa, and then the pupa egg cases, the pupa case that has then closed, so you know what stage that has occurred. And the same for the Drosophila. We provide these websites of the eggs, first instar, second instar, third instar, and then the pupa, and then pupa cases that have opened, as well as some of these items here. And then this could be added in different days. You could actually give different students different days with bags of different kinds of content, and then see if they can back calculate the time period. So that will then allow students to have either kind of a dry lab or a wet lab, and then you can vary those experiments as you choose.